So good evening all. Today I'm going to discuss about apoptosis in detail. So before that, let us see what is apoptosis. Apoptosis is a type of cell death which is produced by tightly regulated suicidal program in which cells destined to die activate intrinsic enzymes that degrade the cell's genomic DNA, nuclei and cytoplasmic protein. So in short, apoptosis is nothing but a programmed cell death. That is, the cell itself will contain some intrinsic enzymes which will destroy the cell's nucleus and its genomic DNA, and also the cytoplasmic proteins also will get depleted. So apoptotic cells, in contrast to necrosis, will break down into plasma membrane bound fragments called apoptotic bodies. This is very important. So apoptotic bodies contain portions of cytoplasm and nucleus. So the plasma membrane will remain intact in apoptotic bodies, but its surface components are altered because to give signals for phagocytes to consume those apoptotic bodies. Apoptotic bodies itself will have some modifications in the surface to give signal to phagocytes for autophagy. Then, as a result, the dead cell and its fragments are rapidly removed, which is very important for a body. So, apoptosis will not elicitate an inflammatory reaction. Okay, that is very important. Apoptosis does not elicit an inflammatory reaction because the dead cell and its fragments are rapidly consumed, rapidly phagocytosed before the cellular contents leak out. So, next, apoptosis is commonly called as programmed cell death because this process is controlled by the action of small number of genes and is required for normal embryogenesis. Even apoptosis is important for normal embryogenesis to occur. Without apoptosis, we can't expect a normal embryogenesis process. So it is a unique mechanism of cell death which is different from necrosis in many aspects. Next, let us see about causes of apoptosis. So apoptosis occurs in two ways: as a part of normal physiological process which gives us a benefit, shows the benefit, and as a pathophysiological mechanism in disease or in diseases in which very different diseases we can observe apoptosis to come. So both apoptosis plays both a positive and negative role in a body. Next, let us see how apoptosis is taking place in physiologic situations. So death by apoptosis is just a normal phenomenon which helps to eliminate cells that are no longer needed. And also it is a mechanism to maintain a constant number of various cell population tissues. See here, if we need our body to maintain a constant number of various cell populations by frequent removal of dead cells by apoptosis, or else it will lead to cancer and other tumor conditions. So there is a two key process: death of cells by apoptosis and their removal of the cells. The debris of the cell should be removed by phagocytes. That is, we learnt previously. So the first one is the removal of supernumerary cells during development. See here, during our development or during embryogenesis, the cell death is very important for formation of primordial structures and remodeling of maturing tissues. Okay, that is very important for involution of primordial structures and remodeling of maturing tissues. Next, involution of hormone dependent tissues and hormone withdrawal. See, we know that during the menstrual cycle the endometrial cell gets broken down during menstrual phase of menstrual cycle, and the ovarian in menopause the ovarian follicular atresia also occurs, and after weaning in the sense okay, so during the menstrual phase of the menstrual cycle the endometrial cells gets break down. So apoptosis is regulated by hormones. So there is a hormonal regulation in menstrual cycle, and hence it will trigger apoptosis, and hence endometrial cells will break down. So during menopause, there is a condition called ovarian follicular atresia. See here, it is nothing but the ovarian follicles will get degenerated and gets converted into corpus albicans. Okay, corpus luteum and then to corpus albicans, and also it will stop the ovulation process. So that is ovarian follicular atresia, degeneration of ovarian follicles. Next, are lactating breast in females. See, after weaning is nothing but giving milk to young ones through the breast. The regression, that is degeneration of the lactating breast after lactating period, that is after milk is given to the baby, the breast starts degenerating. That is also due to apoptosis. Next, cell turnover in proliferating cell population. See here, immature lymphocytes in bone marrow and thymus. What happens is B lymphocytes in germinal centers which fail to express useful antigen receptor. Some B lymphocytes are there. Okay, still there, not all B lymphocytes are get matured. Some B lymphocytes will not express useful antigen receptors, and epithelial cells in intestinal cleft. So how intestinal cleft is formed? This is due to apoptosis. So it will maintain a constant cell number. The other name is homeostasis. Okay, so that is also very important. We need to remove the immature lymphocytes in bone marrow and thymus by apoptosis, and we want to maintain the intestinal clefts through a process of apoptosis, so that constant cell number is being maintained. Next, elimination of potentially harmful self-reactive lymphocytes. Certain self-reactive lymphocytes will cause autoimmune diseases. To prevent those autoimmune diseases, we want the immune reaction. We want process to remove these harmful self-reactive lymphocytes, which will trigger the immune reaction against one's own tissues, which is very fatal. So apoptosis helps in the removal of these harmful self-reactive lymphocytes, as the name indicates. Next, death of host cells that have served a useful purpose. So neutrophils, if you take death of host cells that have served their useful purpose. So neutrophils, death of the neutrophils is nothing but an acute inflammatory response, and lymphocytes are also destroyed by apoptosis process at the end of immune response. See, we want these cells to undergo apoptosis because they do not receive any necessary survival signals such as growth factors and interactions, and it will be a burden for our body to contain all these extra cells. So we want apoptosis to occur. In pathological conditions, we have seen previously physiology conditions. Next, we are going to see pathology conditions. So first one is the DNA damage. So during radiation, when the cells are exposed to radiation and cytotoxic anti-cancer drugs that can damage DNA, either directly or by production of free radicals. All these three terms are important: radiation, cytotoxic anti-cancer drugs which damage DNA with the help of free radicals. In case if the DNA is not able to, if you are unable to repair the DNA, then a cell will produce intrinsic mechanisms that induce apoptosis. In this case, apoptosis will have a protective effect by preventing the survival of the cell's DNA mutation. So we don't want the cell with DNA mutation to survive, because it can lead to malignant transformation and tumor. That is important. Sometimes due to accumulation of misfolded proteins, what happens is endoplasmic reticulum stress occurs, because cell death which is triggered by improperly folded intracellular proteins, and finally endoplasmic reticulum stress response will take place, which we'll be seeing later. Next, apoptosis will be caused due to certain infections, particularly if you take viral infections as a result of virus itself. adenovirus, HIV infections. In these two cases, apoptosis is induced, or in viral hepatitis also. So important host response to viruses. So if any virus is there in a body, it has immediate increases production of cytotoxic T lymphocytes, which are specific for viral proteins, and that proteins will induce apoptosis of infected cells. Infected cells need to be destroyed, or it will spread infection, so that there can also be a significant tissue damage. So this cytotoxic T lymphocyte mediated mechanism is responsible for killing of tumor cells, cellular rejection of transplants, and tissue damage in graft versus host disease. So during transplantation and during tumor, the cytotoxic T lymphocytes will produce certain uh, viral proteins. For that viral protein, viruses contain some certain viral proteins for which the specific cytotoxic T lymphocytes are produced. So these induce apoptosis and helps to destroy the tumor cells and cell rejection of transplantation cells, and especially the damage to the tissue caused due to organ transplantation is due to apoptosis. So apoptosis may also contribute to pathologic atrophy in parenchymal after duct obstruction. For example, if the pancreatic duct is obstructed, then the pathologic atrophy takes place in pancreas. And also similarly, if any duct is obstructed in parotid gland and kidney, then their atrophy also takes place, which is pathologic atrophy. Now let us see some morphological features when a cell undergoes apoptosis. First, in contrary to necrosis, in necrosis we have seen that the cells will get swelled, but here the cell gets shrinked. So cell size is reduced, the cytoplasm is dense and eosinophilic, and then the organelles are relatively normal and are more tightly packed. In necrosis, I said before, necrosis early feature is cell swelling and not shrinkage. That is one important point to be taken down. Chromatin condensation. See, in apoptosis there is a chromatin condensation. So what happens is the chromatin will assemble peripherally under the nuclear membrane. That is, the chromatin material which is in the center of the nucleus will goes to the periphery and attaches to the nuclear membrane into dense molecules of various shapes and sizes. The nucleus itself will break up into fragments. See, apoptotic cell first shows extensive surface membrane blibbing. We have seen what is blibbing in the previous video, and also followed by fragmentation of dead cells into membrane-bound apoptotic bodies, which contain cytoplasm and tightly packed organelles with or without nuclear fragments. That is what we have seen before. Okay, there will be membrane-bound apoptotic bodies consisting of cytoplasm and organelles. Next, we will be seeing about phagocytosis of apoptotic cells or cell bodies, usually by macrophages. So the apoptotic bodies, what happens is they are rapidly ingested by phagocytes such as neutrophils and monocytes and degraded by the phagocytes lysosomal enzymes. That is very important. Next, in H&E stain, has hematoxin eosin stain tissue, the apoptotic cell appears as round or oval mass of intensely eosinophilic cytoplasm with fragments of dense nuclear chromatin. See, and there is no inflammatory reaction that we have seen before. Unlike necrosis, we have seen that there is no inflammatory responses, so it is difficult to detect apoptosis by light microscopy. So that one point is need to be noted down. So that's what as the lesser fragments are rapidly consumed before the contents leak out, and therefore so cellular contents does not leak out, and hence apoptosis does not elicit an inflammatory reaction. Next, let's see about mechanism of apoptosis. So apoptosis is due to the activation of certain enzymes called caspases. Very important one. They are so named because caspases belong to proteases class of enzymes which will contain cysteine in its active site. Okay, They contain cysteine in its active site and cleave proteins after aspartic residues. Okay, so this caspases part is very important. Caspases belong to proteases, and caspases will be existing in our body cells, inside our body cells as inactive proenzymes, that is inactive enzymes, and must undergo enzymatic cleavage. That is what is mentioned. Very important is enzymatic cleavage to become active. So how caspases becomes activated? Through enzymatic cleavage. So the presence of active caspases is therefore marker for self undergoing apoptosis. So if apoptosis takes place, definitely there should be a presence of active caspases. So if it is found in blood serum, then we can confirm that apoptosis is taking place. So apoptosis is divided into initiation phase, during which some caspases becomes active, and an execution phase during which the terminal caspases trigger cellular fragmentation. So terminal caspases that is finally activated to trigger cellular fragmentation, and then we will see that two distinct pathways converge on caspase activation, that is the mitochondrial pathway and the death receptor pathway. Let us see what are these two pathways. So first, if you see the mitochondrial intrinsic, or referred to as intrinsic pathway of apoptosis, it is responsible for apoptosis in most physiologic and pathologic situations. What happens is the mitochondrial outer membrane permeability will increase. Okay, what will happen is, and then there will be release of death inducing apoptotic molecules from the mitochondrial intermembrane space into the cytoplasm. See, the permeability of mitochondrial membrane is increased. As a result, in intermembrane space, pro-apoptotic bodies are present that will induce apoptosis. It enters the cytoplasm. So mitochondria is nothing but these are organelles that contain cytochrome C, a double-edged sword is essential for producing the energy, example ATP. So ATP energy is produced, okay, but it is a function of mitochondria. But when it is released into cytoplasm, an indication that the cell is not healthy, it will initiate. So if these pro-apoptotic bodies reach the cytoplasm of the mitochondria, then it will indicate the suicide program of apoptosis. Or no, actually what I am telling is, when ATP is due to increased permeability of mitochondrial membrane, apoptotic bodies, pro-apoptotic bodies will jump into the cytoplasm of mitochondria, and ATP inside the mitochondria will be leaking in the cytoplasm of the cell. If that occurs, then the suicide program or apoptosis is initiated. So you see, pro-apoptotic proteins such as cytochrome C is a pro-apoptotic proteins. So it is determined by the integrity of the outer mitochondrial membrane, which is tightly controlled by BCL2 family of proteins. Okay, this cytochrome C is under the control of, okay, this control of BCL2 family of proteins. So the family is named after BCL2. So BCL2 is a gene that is frequently overexpressed. What is BCL2? If this is a Viva question, this is frequently overexpressed due to chromosomal translocations and other aberrations in B cell lymphomas. So BCL, there are more than 20 members of this BCL family, which can be divided into 3 groups based on a function. First we'll see about anti-apoptotic. So BCL includes anti-apoptotic. BCL family includes BCL2, BCLXL, and MCL1 are the principal members of this group. They have 4 BH domains and hence called BH14. So these proteins will be in outer mitochondrial membrane or in cytosol or in endoplasmic reticulum membranes. So they prevent, the function of these anti-apoptotic is they prevent the leakage of cytochrome C and other death-inducing proteins into the cytosol of mitochondria. They keep the mitochondrial outer membrane impermeable. Next class is pro-apoptotic. So BAX and BAK are the examples of pro-apoptotic proteins. They contain the first 3 BH domains and hence it is called as BH13. So if it gets activated, BAX and BAK, what will happen is will combine within the outer mitochondrial membrane and enhance its permeability. So unlike anti-apoptotic bodies, in pro-apoptotic bodies, permeability of the mitochondrial membrane is increased. Okay, the precise mechanism by which this BAX and BAK oligomers permeabilize membranes is not settled. Okay. So they form a channel in the outer mitochondrial membrane. It will allow this cytochrome C leakage into the intramembranous space. That's it. So next you have to see about regulated apoptosis initiators. Here examples are BAD, BIM, BID, Puma Noxa, and containing only one BH domain. That is important. So the third, that is which BH domain, if they ask me, should tell us the third of the 4 BH domains, and hence they are called BH3 one-leaf proteins. They contain only one domain. So this activity of these proteins is modulated by sensors of cellular stress and damage. When upregulated and activated, they can initiate apoptosis. Okay. So that is what. So this is enough, I think, for apoptosis. Okay, but we will see it again. So when the cells are deprived of survival signals, they suffer DNA damage or develop endoplasmic reticulum stress. So very important is they usually indication of apoptosis is DNA damage, endoplasmic reticulum stress due to accumulation of misfolded proteins. So BH3 only proteins are upregulated through increased transcription